Uh, my name is Kate Meyer, and I'm an associate professor at Duke University and part of the center that's putting on this event. And so we're going to end this fantastic first day with a panel discussion from some of our speakers from today, as well as some who will be speaking tomorrow. So we have Chen, Pete Dedon, Robert Ross, Dan Fabris, Henning Urlaub, and Ben Garcia, who are going to be joining us. And so we only have 30 minutes or so, but this is designed to be interactive. So just like at the end of the talks earlier today, if you have specific questions or topics that you'd like to be brought up with the panel, please enter them in either the chat or the Q&A, preferably the Q&A. And hopefully we can get to some of your questions and your burning issues that you want to discuss. But to kick it off, I think it'd be great if we could just hear from each of the panelists about what do you think is the biggest challenge that mass spectrometry faces when it comes to studies of RNA modifications? We can go in no particular order. If anyone wants to jump on that one first, go for it.
Sensitivity. Sensitivity is the one that's really holding us up. And it was good to hear Ben talk about nanogram level of input. If we can get down to single digit nanograms, the ability to get tiny amounts of tissue, brain tumor tissue, and then do the extractions and purifications, the amount you're left with is so small that we can't compete with next gen sequencing and PCR to get sensitivity, but we've got specificity. So, yeah, we need better mass spectrometers, more sensitive detection.
Maybe I would add that we probably need some kind of standardizations in the field. We have a lot of great methods, but a lot of them are very different. People are doing all kinds of things. Sometimes it's hard to even compare across methods to kind of see what the losses or gains are. And we just don't have, a really unified standardizations, standards and then standardization methods for QC and everything and the computations. So it's a little bit still wild, wild west in this emerging field.
Wow. Can I throw a quick shout out on that exact point of harmonization and standardization? Pat Limbach, Mark Helm, Viv Chung, and I have been working together, funded on a grant by the Warren Alpert Foundation, to harmonize ribonucleoside analysis by LCMS, all the things we heard about earlier today. And we worked at every step of the process. Viv prepared RNA samples and shipped them to Pat, Mark, and me, so we're shipping to Germany, Cincinnati, and Boston. And every single step of the way, quality control, harmonization, everything, the goal was to let's see how we differ in our results for these three labs that know how to do this well, right? The most troublesome part was getting the RNA from one lab to the next, the dumbest thing you can possibly imagine, shipping it and coming up with ways that didn't harm the RNA, alter the modifications, or just lose due to degradation. So we've got a paper under review at NAR where we've compared, it's all QC metric stuff on LCMS analysis, but I'm with you 100%, trying to harmonize this and get everybody on the same page.
Yeah, that's great that you guys took that upon yourselves to do it. I'd be delighted to see the paper when it comes out, because I think that's absolutely what we need in this field so that we can kind of feel good about all these different techniques that are coming out, that they're going to be robust and reproducible, and so that's fantastic.
I agree. I think this issue really permeates all of our technologies that we have as a field, not just mass spec, but sequencing as well. There's so many different methods to study different modifications, and that was one of the big conclusions from that National Academies report that came out of what we need to fully sequence all the RNA modifications. And it's probably the least sexy aspect of this, but it's one of the most important, because until we have universally accepted standards and methods, we can't make much progress. So I completely agree. Any thoughts from others on the panel in terms of what challenges you think mass spectrometry is facing?
I mean, Henning speaking, for me personally, I ask myself how reliable the software tools are upon all sequencing of RNA, right? I mean, false discovery rate, all these tools, or the twists we know from proteomics. How do they apply? And I mean, I looked at several spectra, and I actually also wanted to show an own MS2 spectra. They are all pretty noisy when compared to, let's say, peptide-derived fragmentation spectra. Yeah, so I wonder, this, in my opinion, this is also a challenge.
Yeah.
Yeah,
So perhaps
definitely.
in
Go ahead.
addition to the sorry, so perhaps I'm thinking in addition to the technical challenges, the thing that really matters is attention from the community. I think many people have been drawn attention to this nanopore and other different techniques. Right now, if there is not a critical mass of people, we will need this technology. So this will be a big problem. So I think, like Ben mentioned, perhaps one day we should write consensus or something together, publish into some prominent journals, draw people's attention, and then when people realize there is a need to do this and there are ways to do it, and the companies can follow up and they can provide service. So and perhaps unify some of the established methods. So that's just my thinking.
Yeah, I completely agree. I mean, and we need money too, right? So we need to get the attention not just of the research community, of course, but of people who can fund our research and even companies, because I think having academia industry partnerships can be really fruitful as well. And I'm glad, thank you again, Stephanie Kaiser posted in the chat a link to the Human RNome project, which is leading this effort to really push this technology and like you said, grab the attention of people that can help fund the research, but also just others in the community, and try and get more of a universal effort behind some of this. So I think that's great.
I think the comment that it really is not sexy stuff that we have to do to do this, there's no chance to get NIH funding, even if we had an NIH. Sorry about that.
I was trying to avoid that, but yeah.
To get NIH funding from a standard study section. So going after Agilent, Thermo, SciEx, all the different companies to try to do that. We've got a couple of small grants from Agilent, but to get all this harmonization done, there's got to be another pot of money to help do this, because it's not cheap.
Absolutely, could not agree more. Any other comments from panelists on other challenges you think that the field will face in terms of mass spec?
I will comment tomorrow on Henning's question about software, and the whole new world and Ben knows this well, deconvoluting fragmentation MS2 spectra from an RNA or DNA. It's a mess. Yeah. Hey, it's doable. That's why there's smart people like Ben who know how to do these things.
Absolutely. And on that front, this was a topic that, has come up in the past. I'm curious to hear the panel's thoughts on, the potential role of AI in that process, and how that might really facilitate developing better methods. Is that something that you all have started to incorporate, or, what are your thoughts on that?
It's garbage in, garbage out as far as machine learning. And you really have to establish the baseline as what we're discussing right now before you start feeding data in, or you're going to go off on a tangent that could be devastating later.
Just use the Google AI now to ask a question on RNA modifications, and you get such embarrassingly bad answers, right? Because there's just too much garbage out there.
Yeah. I mean, I can't even get, Dolly to make me an image of RNA half the time. It just wants to send me DNA. So,
yeah. One thing that's challenging too, I mean, especially now that, sensitivity is getting better. Everybody's, getting better at doing these, there's going to be more modifications identified, and and things like this is it's just being able to validate them by creating, the synthetic version to make sure your MSMS and your retention times and everything looks good. And I think that's a big bottleneck, the synthesis of some kind of very exotic modification, weird thing. So we need definitely, chemists to get involved in this too.
And I
Yeah,
think,
I would like to point out one other thing as far as normalization. Like I can look at everyone's protocol, and we all seem to follow the same type of extraction procedure, where that's using some type of guanidinium denaturant and phenol chloroform extraction. If you look, I think Mark Helm had a paper with Yuri Maturin a few years back where they said, it's the effect of looking at the data that we got. We don't even know if that's what's really happening in the cell, because in the whole process of extracting it, we could be changing it. And no one's I haven't seen it in a long time where someone would use like a French press or even just enzymatic degradation and then just compare, with column chromatography and maybe an alternative digestion scheme, and just compare to see if the modifications that we're seeing are really what we're looking at. So there is just a lot of gray area going back to the very beginning, from cell to data. I mean, every step of the way is we've just, this worked, and we got our data, we got our paper, we got our grant, we developed our instrument, and nobody's really looked deeper into it because it just seemed to work, and nobody wanted to go back and look at it. So going back to the basics, I think, is going to be very important to establish this baseline, like what's really the normal levels of modification. Who says that hex cells or HeLa cells are really the platform to base anything on?
Stephanie Kaiser wrote a great accounts of chemical research piece last year on pitfalls in the whole RNA modification analysis realm. It's LC-MS analysis of ribonucleosides. It's not the sequencing part. Someone else should be writing one of those soon. But really pointing out some of the things you mentioned to that exactly. The pitfalls of using different kits and types, and just a subtle change in the pH on one of these Qiagen columns, the 5S will fall out of the small RNA pool and into the large. And just the amount of confounding variability that you can get from something as simple as isolating the RNA. Very, very much agreeing with you.
Yeah, it's not a simple problem. Yeah.
So, another thing that came up today, I think especially in Kristen Kuma's talk was, the strength of being able to do things that don't require, bespoke methods and equipment and things like that. And really it speaks to what we're talking about here of getting, more widespread adoption of standards and practices and things like that, but making it accessible to every lab, right? So being able to use core facilities to be able to analyze what you want to analyze. And I think that's critically important as well. So I'm curious, what the panel thinks about, what can we do to get there, to get more widespread adoption of some of these methods in a way that's compatible with most core facilities that are at, various universities and institutions. I mean, I know it might not be a one size fits all answer because everyone's studying different things, of course, but just curious to hear how we can bridge that gap, because I think it is a critical one, right? We can't get widespread adoption of this, and we can't move forward unless we have lots of people who can do this easily, right? It was the same thing with next gen sequencing, a couple decades ago, and eventually we got there, right?
So I would like to address what Kristen said about the ion pairing and HeLiC and the core facilities, because I came from a core before I joined Thermo, and we had fit for purpose instrumentation, and a lot of it was inherited because somebody had bought it for a specific purpose, and it was left over and it was being utilized. nucleic acid mass spectrometry is not going away. It's only going to grow. Here, we have an Ascend tribrid in the lab here that I share with a proteomics expert. And what we found is that you need a separate pump. You don't need a separate instrument. The way I believe that it's been explained is that the ion pairing reagents get into the degasser of the pump, and it doesn't want to come out. So as far as capital expenses, a core really would only need a pump, because if you use just standard cleaning procedures, it's not going to contaminate and affect the sensitivity of your mass spectrometer. You just have to switch the pump out, and a pump's a lot cheaper than a high-end mass spectrometer. And if you're going to have this type of analysis in your core, you should be able to offer the best sensitivity you can for your clients. The researchers they're going after the grants, and the best sensitivity is with ion pairing.
If I may interject, there is a whole lot that we can do on the hardware side, but I think we are still lacking in terms of education. There's a lot of facilities where they have no idea on how to run nucleic acids in general. And the first time you approach it from the literature point of view, the phases are different, the sample preps are different. There is a whole lot that, your run-of-the-mill facility, they are used to do metabolomics, proteomics, and now they have to switch completely. Best results, at least for me, are in negative ion mode. When I mention negative ion mode, people look at me like, you have an accent, you are green, you must be from Mars. They don't understand the idea. And just having an instrument in your facility dedicated to nucleic acid analysis in negative ion mode with the proper phases, that will already make a big difference in terms of making it available to people in that department, having that capability.
This gets to what Stephanie's organizing in Frankfurt coming up, this workshop, outreach. And again, it's not sexy and nobody pays for it, but it's kind of up to us to get out, our students, our postdocs, us, to get out and do demonstrations to our core facilities at our universities on how this works. We're trying to develop things that any core facility with an Orbitrap or a similar instrument can hook it up. Ion pairing agents are foul, dirty words in my lab after somebody did use it and we had the instrument down for months. If I think trying to balance now accessibility, sensitivity, compromising a bit, to get any university core facility, any company with a mass spec should be able to do this, and figure out some way to get a protocol in someone's hands. How do we do that as a community? A standardized, reproducible, robust, stainless steel protocol in everybody's hands.
Completely agree. Yeah.
But then I would really like to thank you to raise this point. Yeah, this is exactly what we are confronted with. So I have also a core facility running, but we are highly specialized in proteomics, right? And we could easily dedicate an instrument, right, to negative mode, also for the LC settings. And then comes the next problem, lacking co-workers, that are dedicated to do this, because you have to establish this and takes some days, weeks, and then you have to work on it. And this is the next thing, definitely. But it has to be done. And then you're completely right, Peter and Rob, that if there's an instrument available, it can be done and it should be done, because it's even at our institute, it's requested more and more, definitely.
It's almost going to be, I don't want to sound like an old man, which I am, but it is going to be generational from the standpoint, number one, of taking proteomics facilities now and convincing people we're not going to mess up their instruments. It's okay to change over. We'll show you, no problem. And then number two, getting the demand up at our schools, companies to get a queue going to want to do that. Because if there's interest, if there's a line outside the office, schools will accommodate that. Need drives availability. So it's going to take several years to enculturate our core facilities on how to do this. And we need a package. Maybe Stephanie should come up with a giant book, a playbook on how to get this done at a school, and she can distribute that to our school. Sorry, Stephanie, former postdoc, I can abuse you here like that, right?
Am I not doing enough yet? One step at a time, okay? All right. Yeah, we have to do that.
Yeah, could not agree more. So I know we've got just less than 6 minutes here. So I think it'd be great. You guys are all, the experts. It'd be great to hear from each of you. What do you think? What does the next 5 years look like for you? Either what's the low-hanging fruit you think we should go after? Where do you think this technology is going to go? Or maybe just what's your wish list of what we think we could achieve within the next 5 years and how we can get there?
Enzymes.
Or beyond 5 years. It doesn't have to be 5 years, but
yeah, enzymes. That's my wish list.
Second that motion.
Yeah.
Sorry, Rob, did you say enzymes?
I did, yes.
Okay.
Yeah. We need for RNA what we have for DNA. Yeah.
Yeah.
And better software, especially if you want to shotgun stuff. NASA is great already, but it has its problems with certain types of analysis of data. But it's being developed, right? And it's going to be there. And in 5 years, I think we have something like MaxQuant available for RNA. But for that, we need data to feed into the software pipeline, right? So we need to have also the data to push the software. I think this together with the enzymes, as Rob says, and then I think we have a great future, very similar to proteomics ahead of us. Sorry for chiming in.
no, that's great. Love hearing your perspective.
No, this is definitely what we need. Yeah. And then you can really go for the full, as you told, RNA-ome, yeah, to sequence every mRNA that is transcribed, definitely, yeah. Also at various stages of processing would be fantastic. And it's doable, definitely, as so mass spec has the power, yeah.
So I hate to come at it from the vendor point of view, but the biopharma finder oligonucleotide sequencing is very good. Internally, I use it to map modifications in tRNA digest all the time. I've mapped modifications in ribosomal RNA digest, and while they're small, the coverage is excellent. And I know that for academics who are mostly here, sometimes that's not an option. We want an academically driven or freeware type of software to answer the question. But Compound Discover for nucleoside analysis and Biopharma Finder for oligonucleotide analysis are two very powerful software options. And if you could purchase them and put them on a centralized computer at your core, like we had in Cincinnati, where anybody could come and use it, you could train them, you would see the benefit of using this software. I mean, it's very good. I work very closely with both the software teams, and it's a very good software. They turned me into a believer within just a matter of months from joining. And I didn't get paid for saying that.
So you also feed it with a FASTA file that contains what? 500 different sequences, and it finds your fragments.
Not to go too far off the trail, but you put your sequence in. You put your sequence in that it's there. Like say we were going to map phi, and you choose your enzyme that you use to digest it, and there's a list of enzymatic digestion options. and you just import your raw file, and then it processes it. It can find sodium adducts, it can find truncations, base loss. It's very powerful, and like I said, I work with that group, and I'm constantly feeding them data and showing them what, this is real life, and a lot of it's market driven. A lot of our customers are startups here in Boston that are doing therapeutics, but I come from the academic background as well, so I'm constantly having them work on the biologicals, and it's very nice, and I can show it at the workshop, Stephanie.
Where do you all send your sequencing data when you're ready to publish? What public database, which we all have to do now, deposit our raw data into the database? What database are you folks using? I'm just curious because I think we need a database dedicated to RNA sequencing, which is not proteomics.
You mean these RNA modification data sets? You mean
sequencing data, raw mass spec sequencing data, any kind of RNA modification, RNA data?
Sequencing data, we usually deposit in NCBI, so there is a GEO. But for when we publish, there are separate requirements for RNA modification. We just share to figure share. I'm not sure what is that, but that's what we are asked to do. So to deposit the raw data into figure share. So that's what we did in the previous publications for all these modification data.
So if it's just sequencing data, we can go to standard sequencing
Yes, I think
databases.
The NIH, they usually accept this GEO. So for RNA sequencing, DNA sequencing, but for modification, it's a little bit different.
I agree that I think we need a centralized, database, and there are some, existing, of course, RNA modification databases, but the vast majority of them are, developed by certain labs, and then, the postdoc might leave or whatever, and they aren't necessarily maintained. So I think we need, like Modomics and, centralized where, because we do the same. We put our data right in GEO with NCBI, but I think we definitely need a centralized, curated, updated database like that so that we can incorporate mass spec data, sequencing data, all of this, which again exists, but we need to make sure that it has the resources to continue to exist and to be maintained, and not just have all these smaller offshoots that are just maintained by single labs. Okay, so I know we're at time, but if there are any kind of last thoughts from the panelists, feel free to share them now on anything.
It's an exciting time to be doing this. It's Wild West in many ways, but boy, it's the amount of cool new stuff coming out of this, the RNOme and modification analysis, just mind blowing. It's wonderful.
Absolutely, yeah. I think, yeah, that's maybe a great way to end, because I think we're all in agreement there. This is a great time to be in this field, and I think sky's the limit on what we're going to be able to achieve. But like we talked about, there are a lot of challenges to get there. Okay, well, I want to thank all of you. This is a great panel, and thank all of our speakers today. I think this was a great first day. I definitely learned a lot. And to everyone else, we will see you tomorrow morning. We're going to start again at 10 a.m. And I think we'll have one of our panelists. We'll have Pete Dedon as the first speaker tomorrow morning. So thank you, everyone, and enjoy the rest of your day. We'll see you tomorrow.
Thank you.
Thank you very much.